you guys hear me? Okay. Well, it's 6, so I'm gonna get started. Uh, so before I start, I'll start with an introduction. My name is James. I'm a 9th grader from North Allegheny Intermediate High School, or NAI, and my favorite subject in school is biology. And today I'll be talking about biotechnology. Okay. So first, to talk about biotechnology, we first need to start with what is biotechnology. And so biotechnology is a field where you can just read on screen, biotechnology is the manipulation of organisms or their components to make useful products. And a subset of biotechnology would be DNA technology, which is using DNA tools to analyze gene expression. So essentially, biotechnology is a field that specializes in using biological systems and organisms to develop useful products and technologies. It has like a bunch of applications, some in healthcare, agriculture, environmental conservation, and others. It has revolutionized medicine. It improves crop yields, and it helps make things like GMOs. It helps with biofuels, and it helps with different industrial processes. And more specifically with DNA technology or DNA tools, this has helped revolutionize genetics, genomics, forensics, and biotechnology. It uses techniques like PCR, polymerase chain reaction, DNA sequencing, cloning, and gene editing in order to change DNA to make it something that is useful for humans like you and me to use. And it has a lot of applications across different fields like diagnostics, genetic testing, DNA profiling, and creating or like genetically modifying organisms. So, all in all, pretty useful. Okay. So, okay. So before we start talking about like DNA technology and everything, it's good to talk about the history of it first. So the history of like DNA and everything basically started with Darwin, who like most people know, he discovered like natural selection, evolution, all that good stuff. Um, so he traveled the world on the HMS Beagle, and he visited the Galapagos Islands, and on the Galapagos, he noticed that finches had different beak sizes because of a drought that's it's like it made seeds different, and they had to adapt to the seeds on each of their islands. This is called the bottleneck event, by the way. And even though he didn't directly know about DNA, he basically kind of like established the basics for DNA in the future. He had a theory of evolution, and this laid the groundwork to understanding how species adapt and change over time. This made a framework for future scientists to explore like DNA and how it is carried on among other organisms. And then later, like a few years later, Gregor Mendel, did some stuff. He was a monk, and he did experiments on alleles and allele frequencies, all that good stuff. He had a lot of, work on inheritance, and this is known as Mendelian genetics. He laid the groundwork for the discovery of DNA because he discovered that there's certain traits in organisms that can be carried on to their offspring. And this, and he didn't know it at the time, but this thing that carries it is actually DNA. And then, so we move on to the experiments of Oswald Avery, who did something pretty cool, as so did the rest. So he basically explored how DNA is carried through different organisms. So, for example, he like experimented with these rats, right? Uh, he discovered that if you take a rat and give it pneumonia, and give it a pneumonia, it's going to die. And if you take a rat and give it something that's not dangerous bacteria, it's not going to die. And if you give that same rat uh, a heat-killed strain of pneumonia, the rat is not going to die because the pneumonia is dead. But if you combine the heat-killed pneumonia and the like non-hostile bacteria, the heat-killed pneumonia is going to give the not dangerous bacteria its genes and make it dangerous, which will kill the rat. So he discovered that like by creating a substance that is extracted from the bacteria with an enzyme that destroys proteins, lipid, and carbohydrates, it shows that DNA can change a bacteria that's not dangerous into a bacteria that's dangerous and can kill. They still didn't really know about DNA at the time, but it shows that DNA can carry, or there's something there that can carry genetic traits to other organisms or the offspring of organisms. And then a few years after that, Rosalind Franklin, who, a lot of people know, she photographed the structure of DNA under Maurice Wilkins. So she just confirmed that DNA had a double helix structure, which is important in the understanding of how DNA works. And then later, there was Watson and Crick. And what they did is they compiled work from a bunch of different people, namely Chargaff and Rosalind Franklin. So Chargaff, spelled Chargaff, he basically established that, ATGC, so A, adenine binds to T, thymine, and G, guanine binds to C, cytosine. And Franklin, as we just discussed, she found the structure of DNA itself. So they compiled this and they made the first accurate model of DNA. So DNA has a double helix structure, and it's, the adenine binds to thymine, cytosine binds to guanine. And then later on, after the work of Watson and Crick, then this guy named Frederick Sanger came along. And what he did is he developed something called Sanger sequencing or DNA sequencing. And this technique allows for accurate determination of the order of nucleotides in DNA, and it helped revolutionize genetics and biomedical research. Essentially, if you simplify it, what this means is he developed a way to look closely at each of the nucleotides, which would be ATGC, adenine, thymine, guanine, and cytosine of the DNA, and you can look at it and basically see what the DNA is made of, or the structure of the DNA. And then shortly afterwards, the Human Genome Project began. And this project basically is the goal is to map out the DNA of a human. So you just want like the complete genome of the human, which is really long. It's like billions of base pairs long. And the first human to get their genome mapped out was actually one of the co-discoverers of DNA, which would be James Watson. And in 2003, the Human Genome Project finally was completed, where Watson got his DNA mapped out, which is pretty cool. And later on, we continue to learn stuff about DNA every day, and a bunch of cool stuff. So now I'll actually talk about some biotechnology. Okay. So the first important thing about biotechnology is nucleic acid hybridization. If you just read what's on the screen, nucleic acid hybridization is the base pairing of one strand from a different nucleic acid molecule. So it's basically a process where complementary DNA or RNA bind strands together. So if you look at the picture, basically something like helicase is going to open the DNA. Okay. So basically, this is a DNA probe, and then something called helicase is going to open the DNA. It's just an enzyme or a protein whose role is to open the DNA, so like this. And then this DNA probe is going to be treated to a lot of heat, which is going to denature it and force the two strands to separate like this. And then if you insert these two strands into the DNA, which is going to be opened, you can basically make another strand of DNA for this strand. So basically the base pairs of this strand is going to they're going to change so that you can bind to this strand here, and the structure of this strand is going to change so it binds to this strand. So basically, nucleic acid hybridization is a process where complementary DNA or RNA strands bind together. It is used to detect specific sequences and to study genes. So by bringing together matching strands, scientists can look at the structures of nucleic acids, and this can help with different things like genetic, can help with disease diagnoses too. And something that's important to note is what complementary DNA or RNA is. And if you look at, oops, my bad. And if you look at it, this is going to be complementary DNA, also known as cDNA. It is a single-stranded piece of DNA. So just DNA is a double helix normally. It's going to be basically like ripped apart, and it's going to turn into one strand. And this single strand of DNA, it can pair up with another strand of DNA. In the case of nucleic acid hybridization, it will pair up, for example, this strand here is going to pair up with this strand, and the base pairs are going to change, but they're going to hybridize, which essentially the base pairs. So, for example, if there's an A, the base pair, the corresponding base pair on this one is going to change to a T because A binds with T according to Chargaff's rule. And the binding of complementary DNA strands to another strand is called nucleic acid hybridization, which is the title. Right. So something that's really important in biotechnology is specifically DNA sequencing. And there's a bunch of ways you can sequence DNA. They're here. Sanger DNA sequencing is the oldest one. Massively parallel DNA sequencing is slightly younger. It's developed pretty recently. And then Nanopore is the youngest one. And you can see they're all different and they all excel in different areas. So I'll start by talking about Sanger DNA sequencing. So Sanger DNA sequencing, which is here, is a traditional but a more slow way and more expensive way of sequencing DNA. And even though it's like slow and expensive, one of the pros to it is that you can use it to read long DNA strands, and you can read them with really high accuracy. So if you have like a really long strand, like for example, the Human Genome Project, you would be able to look at the long strand and you'd be able to read it with high accuracy, so there'd be little mistakes made. This one here is massively parallel DNA sequencing. It's fast, it's not expensive, but it doesn't read these long strands really often. And also, if you give it like a really long strand, it's not going to be very accurate. So And then there's also Nanopore DNA sequencing. This is a more portable version, so you don't have to use like these huge machines and labs to do it. And it offers sequencing in real time. So normally for Sanger and massively parallel DNA sequencing, you need to like send them to like large lab or something else, and you would need to wait like a week or a few weeks get the to get the structure of the DNA. But for nanopore DNA sequencing, you can do it yourself in your own lab. But but the difference is that it's not as accurate. So it's kind of like massively parallel sequencing. It can do it pretty fast, but it's not as accurate. But the pro is that it's portable and you can do it yourself, and also can read longer lengths. So yeah, now I'll talk about like each DNA sequencing thing in depth. So first with Sanger sequencing. Sanger sequencing was developed by Frederick Sanger. We talked about him in the Some History thing. It determines the sequence of DNA. It involves DNA replication using a DNA template, DNA, DNA polymerase, and specific primers. During the DNA sequencing product, small amounts of fluorescently labeled chain terminating nucleotides, also known as dinucleotides, are added to the DNA replication reaction. And these chain terminators, they don't have the 3-OH group that's on every DNA. And you can basically add a bunch of nucleotides to the base because there's no ending cap. And then as a result, you can essentially grow the DNA using DNA synthesis. And the terminated DNA fragments that you produce are going to be separated using something called gel electrophoresis, which separates the fragments based on their size. And then you can detect the fragments using something called fluorescence, which is you can label each nucleotide. Like for example, A, T, G, and C are going to be different colors. And then you can give each one basically a different color and then analyze the structures based off of that. So, and then after you analyze the colored bands, you can read DNA sequence, and then the DNA fragments are read from the smallest to largest, representing the sequence of nucleotides in the original DNA molecule. So, if that sounded complicated, that's fine. It's a little bit complicated. So, essentially, what you're doing with Sanger DNA sequencing is the first thing you're doing is you're synthesizing new DNA, and then you're using something called fluorescence to read it. So you're going to basically use nucleic acid hybridization. You can use something called a probe, like DNA molecule kind of, and you can add that DNA molecule to your original DNA molecule that you want to read. And then when you do that, you can synthesize a new sort of DNA molecule. And what this does is it allows you to analyze the DNA molecule using something called fluorescence. Essentially, you're going to give each molecule a color. So there's specific markers that you can use that bind to, for example, A adenine. This is a specific marker that binds to that and can turn it a specific color, like for example, red. And there's also G, C, T. There's all markers that bind to them and give them their specific colors. So you're going to separate the DNA molecules. You're going to fluoresce them, essentially bind molecules to them to give them color. And then after you do that, what you do is you can use something like you see on screen like this, and then you can read the DNA sequence. So you see that there's a marker at each line, and then this allows you to read the sequence. So you can see here the DNA sequence would be this, B, A, G, T, T, G, C, so on. And it allows you to read really long lines because it's not very difficult to do this, and you can read really long lines with pretty high accuracy. But it's just really expensive because it takes a lot of resources to do something like this. And then the next sort of DNA sequencing type is called massively parallel DNA sequencing. It's also called next-gen sequencing or NGS. It's an approach that uses basically you're sequencing millions of DNA fragments at the same time, which allows you to go really quickly since you're not sequencing them in a straight line. So like for example, Sanger sequencing, it would be you have a really huge DNA, like a huge DNA molecule, and you're sequencing it one at a time. But for massively DNA sequencing, you would be splitting it into different fragments, and then you'd be sequencing each fragment at a time, and then you put that fragment together. You put each sequenced fragment together into a really big DNA molecule. And the good thing about this is that it's fast because you're sequencing them all at the same time as opposed to in a linear like kind of progression. But the con to this is that you're splitting the DNA molecules up and then you're putting them back together. And there's a margin of error that comes with doing this. So essentially, you're trading off quickness or efficiency for a lower accuracy. Because when you're putting when you're putting the fragments together, there could be some error there, which leads to some possible errors in reading the DNA. So it uses essentially programs or platforms to generate a lot of sequencing data in a short amount of time, and it basically splits it off into small fragments, and then it can decipher each fragment, and then this allows it to be scalable and cost effective, but it trades off for accuracy. And then the last one is nanopore DNA sequencing. So this this type of DNA sequencing is a relatively new type. It's the newest type out of the three here. It involves passing DNA through it involves passing the DNA molecules through something called a nanopore, which is just a tiny hole on the membrane of the on the membrane that they give you. So if you look at the image, this tiny thing here is going to be the nanopore. You can pass the DNA molecule through the nanopore, and then as you move through the nanopore, an electric current is going to be detected, and essentially you can read each unique nucleotide passing through the pore using this electrical current. And this allows you to do it in real time, so you can see each base pair as you're passing the DNA molecule through, and this allows you to do it really quickly. It allows it to be portable, so you don't have to send it to a huge lab. You can just do it in your lab where you are, and it also allows you to do it it's pretty simple. So you don't have to use any of the complicated machines involved. In the other two, you can just use the kits that you're given. This allows it to help in the field of genomics research. It allows for quick and portable diagnostics, and it allows for field work. So, for example, if you're not in a lab, if you're outside, or you quickly get a sample and you need to analyze it, you can just bring it quickly indoors or something to a sterile environment, and then you can use nanopore DNA sequencing to look at the structure of the nucleotides just really quickly. So you can see in this image, right, there's the DNA that's passing through the nanopore, and then this electrical current there's an electrical current running through the nanopore, which reads each nucleotide sequence as you're going through. Okay, wait. Okay. So the next thing we'll talk about is DNA cloning. So DNA cloning is a technique that allows you to create multiple copies of a DNA fragment. And this involves several steps. the first step is you take a DNA fragment and you isolate it from the source of the DNA. Sorry. So yeah, so essentially you just extract DNA from, whatever organism you want, and then you would take a vector, such as a plasmid, and you would select it to carry the DNA fragment that you have. So if you look at this picture, this target gene is it's the DNA you extracted from the organism, and what you want to do is you want to clone it. And this is the plasmid here. Essentially, all the plasmid does is it carries the DNA. So you can use something called a restriction enzyme to essentially cut the DNA out. You can cut a segment of DNA out of the plasmid, which is here, and you can insert your target gene or the DNA that you get from the DNA that you want to clone into the plasmid. And what this allows you to do is you can essentially create something called recombinant DNA, which is DNA from two different organisms. And then essentially what you can do now that you have the recombinant DNA is that you have a plasmid, and then you can clone that plasmid, because the plasmid is like a full sort of DNA molecule, but the target gene is only like partial, so it can't clone itself. So essentially what you're allowing yourself to do is you're allowing yourself to, you can clone this plasmid with the target gene inside of it as much as you want. And then you can use restriction enzymes again to cut out the target gene multiple times. And then now you'll have multiple target genes. So this is the end result of joining the target gene with the plasmid. And then after you clone the plasmid multiple times, you can cut out the target gene and you have multiple sort of copies of it. So DNA cloning, pretty important. It's revolutionized research in the fields of genetics, in the fields of biology, in biotechnology, and it allows you to study gene function more accurately. It's helped with a lot of things, like it's played vital roles in making different pharmaceuticals. It helps with the creation of insulin and different growth hormones. And it's also helped with creating GMOs, which are just, it stands for genetically modified organisms. They're basically organisms that you can edit using DNA cloning and other methods to make them more like resistant, or you can make them like reproduce more or make more meat, things like that. So in addition, it's also helped with things like diagnostics, forensic analysis, and it also helps with more understanding of genetic diseases. So like whenever you don't have a lot of DNA, you can use DNA cloning to clone the DNA, and then you have more of the DNA that you can study more easily. Right. And then the next thing I'll talk about is gel electrophoresis. I talked about it a little bit in DNA sequencing, but I didn't explain it a lot, so I'll explain it here. Essentially, gel electrophoresis is a technique where you can use it to separate DNA, RNA, or proteins, and based on their one, size, and two, charge. You essentially place molecules in a gel matrix and then apply an electric field to that matrix. And this allows molecules to migrate through the gel based on their charge, with negatively charged molecules moving towards the positive electrode and positively charged molecules moving through the negative electrode. That makes sense because, opposites attract. And then as a result, the molecules are going to separate into different bands or regions in the gel. And this allows you to analyze and visualize essentially the build of DNA, for example. This is used in like a bunch of different fields, like molecular biology and genetics, forensics, etc. So if you look at this picture, you can see the structure of something for gel electrophoresis. Everything starts at this power source here. This power source basically can apply you can apply charges to different ends. So you can see there's a cathode and an anode. The cathode gives a negative charge and the anode gives a positive charge. And so basically you can place your DNA, RNA, whatever you want to analyze onto the gel, which is this in the middle here. You put it in the wells more specifically, and then you can turn on your power source, which is here. It'll produce a negative charge at the cathode, a positive charge at the anode, and then and then your DNA, RNA, whatever you want to analyze, is going to move towards one of the ends, and then this allows you to create different bands. So if you if you put something in this machine and ran it, you would see a bunch of different lines, and this would allow you to read the DNA based off of the position of those lines, and it allows you to see like the size and the size of the molecule, and allows you to see exactly what is made of whatever you're looking at. Right? So we're at a halfway point right now. I think it's good to take a little break right now, since I've been talking a lot and everyone's probably a little tired. I think we can take a 5-minute break right now. Right, I'm going to use the bathroom. In 2 minutes, I think we can wait another 3. It's been 5 minutes, so I'll start talking again. All right. So the next thing we'll talk about is PCR, or polymerase chain reaction. This is something that allows you to amplify a specific DNA sequence. So it's kind of like cloning, but you can copy like a specific fraction of DNA within a larger sequence. You can copy a sequence that's slightly smaller within that large sequence. And essentially, this involves 3 steps: denaturation, annealing, and extension. So for denaturation, the DNA template is heated to a high temperature. Normally it's 95 degrees Celsius, pretty hot. This causes the double-stranded DNA to separate onto 2 different strands. So you can see in this image, this would be the denaturation. So you're basically just denaturing, it kind of makes sense. You're just heating it like really hot to separate the DNA into 2 different DNA like fragments. And then the second step would be annealing. So in annealing, you lower the temperature back down, and you're using something called primers to bind to the complementary sequences that are flanking the target DNA region. And then you're using primers, which are shorter DNA sequencing things, which basically start as points for DNA synthesis. So essentially, what you see here is, this step is the annealing step. You're attaching these orange things, and these would be the primers. So you would attach them at the 3' end each time, because DNA sequencing always starts from the 3' end and then goes to the 5' end. And essentially, a primer is just a short sequence of DNA that binds to the longer DNA sequence for synthesis, which is the 3rd step. It's called extension. So basically, you're using DNA polymerase to synthesize new nucleotides, and you raise the temperature again to let the enzyme to synthesize new DNA strands, and you use the primers as a template. This allows you to replicate that strand of DNA. So essentially what you're doing here is you have the primers. The primer is just a short strand of DNA, and you're going to use that strand as your starting point. So essentially everything with the DNA polymerase, and what DNA polymerase does is it synthesizes new strands of, or it synthesizes new nucleotides that are complementary to the original one. So if there was an A here, the DNA polymerase would synthesize a T because A binds to T. And if there was a C here, the DNA polymerase would synthesize a G because C binds to G, because ATGC. And essentially it would add these new nucleotides to the primer, which is basically just the starting point for the DNA synthesis. And what you see is that you would basically, there's 3 steps, and you would do this like a bunch of times. You would do this normally 20 to 40 times, which makes, makes 2 to the power of 20 or 2 to the power of 40 different DNA molecules. And you just do them a bunch of times, and then you can use this to amplify even a small DNA segment. So for example, if you're doing an experiment and you're looking at, for example, a patient's a sequence of patient DNA, and there's a small segment of DNA that you think is causing a certain disease, and you want to test it, but you don't have a lot of that DNA, you could use PCR to test that. So you denature it, you anneal it, and then uh, extend it using DNA polymerase. And then what you would have is you would have basically like thousands, millions of copies of that small piece of DNA that you could now use to analyze. And this helps with things like diagnostics, like I just said. It helps with like forensics, and it helps with like DNA sequencing. So, all in all, pretty useful. And then the next thing is RNA sequencing. So I talked about like DNA sequencing, all that stuff. RNA sequencing is a little bit different. So there's a few things that are important in RNA sequencing. So the first thing is RNA sequencing. It's also known as RSeq, RNA-seq, and it's used to determine the sequence of an RNA molecule. So it allows you to read the molecule, and like just to specify RNA, uh, instead of T, thymine, it uses U, uracil. So instead of ATGC, it'd be AUGC, where U binds to adenine. So this involves RNA sequencing. It involves several steps. The first step involves RNA being isolated from a sample and then converted to something called complementary DNA using reverse transcription. And this sounds kind of, complicated, but it's not too bad. So if, all right, so basically what you'd be doing is you have RNA. RNA is single-stranded. It's like a line. And then you draw, see here. You have a strand of RNA. This is your RNA. And essentially what you would be doing is you would be doing something similar to what's used in PCR. You'd have a primer here, and then you'd use a polymerase to add different nucleotides that correspond to what you have here. So you would have like a primer, and then you would add a bunch of different nucleotides to change it to complementary DNA. And what complementary DNA is it's a synthesized DNA molecule that's derived from RNA. So it's a single-stranded, meaning it's not a double helix. There's only one strand, and you can convert your RNA into this kind of DNA. So, like for example, just to make it more clear, for example, if you have a U here, according to Chargaff's rule, U would be A, because U binds to A. So you'd essentially just make a complementary DNA that can bind to this RNA. And then if there's a T, or T is not possible, my bad. If there's an A, this would be T, because this is DNA and not RNA. So there'd be a T here. And then if there's a C, it'd be a G. If there's a G, there'd be a C. So it's synthesized like that. And then now that you have this strand of cDNA or complementary DNA, you would amplify it. You could use like PCR, DNA cloning for this. You would amplify it, and then you would sequence it using different sequencing technologies, things like that. And then the resulting sequence would be aligned to a reference genome or transcriptome, and then the abundance of each DNA molecule would be quantified. Essentially, what you would be doing is, if you look at this, this is the RNA. This is the RNA. You convert it to cDNA here. And then you have a genome here. And you have a genome right here. And what you can see is you're going to line up your molecules you're going to line up your molecules so that it matches up with what the genome is. And then you're going to move out specific nucleotides in order to easily read what you have just assembled. So essentially, RNA sequencing is pretty important. It's used in a bunch of things like genetics, genomics, and even research. You can use it to find biomarkers, study processes, etc. The strength of this is that you can use it to read RNA sequences, and you can use it to basically see what's happening in different reactions based off of the RNA sequence. And then moving on, the next thing is the CRISPR Cas9 system. So if you don't know what it is already, it's pretty famous. It's something that was discovered very recently, 10 years ago, maybe not that recently, pretty recently. And it has been pretty important in new biotechnology. So it's been a big step in making new and novel biotechnologies. So like CRISPR-Cas9 is not like a process that was developed by humans. It existed in bacteria as a defense against bacteriophages. So essentially, bacteriophages are they're viruses that feed off of bacteria. So they can do this thing, right? They can do this thing called the lytic cycle. It's where they insert their own DNA into the DNA of a bacteria so that they want to reproduce. And if you put your DNA in the bacteria, that counts as reproducing because the bacteria is going to reproduce, and then your genomes are still going to be carried on. And this isn't good for the bacteria because you're essentially just cutting out like genome that they probably need, and they still want like their genome because it's important to them. They like evolved it and they probably need it to do basic things. So CRISPR-Cas9 is something that they can use to cut out the DNA of a bacteriophage. So it's their defense system in bacteria. It uses something called restriction enzymes, things like that, and it uses also like STRs, things like this. And you can cut out the bacteriophage DNA, and then you can use something called SNPs or SNPs to insert your own DNA back in, or you can use it to repair it. So basically, like how do humans use this? So CRISPR-Cas9 allows scientists to make precise changes to DNA within an organism. It helps with something called gene editing. It allows you to change the gene of an organism. So CRISPR, and there's CRISPR and there's Cas9. CRISPR is this part of the name. It's the short repetitive DNA. It's basically a bunch of short repetitive DNA sequences, and Cas9 is restriction enzymes. So essentially, the way the system works is Cas9 is going to bind to a target sequence that you want to cut out and replace, or that you want to cut out and change. And essentially, Cas9 is like molecular scissors, if you can imagine it like that. So it cuts out a certain sequence of DNA, and it uses a guide, which is just an RNA molecule. And then on the target site, the Cas9 is going to cut out the DNA, and then it allows you to change that DNA by however you want. So you can insert your own DNA, or you can just cut it out and remove it. So, like, what are the impacts of biotechnology, DNA technology, all of that stuff on like humanity, and how does it affect humanity? So, it affects it in a few key ways. So, one of the things is it helps with advancements in medicine. This is pretty self-explanatory. DNA tools, biotechnology, they've revolutionized medicine, and they allow you to do more diagnostics allow you to do more treatments, and they allow for more targeted therapies based off of the genetic information that you can gain. So for example, if you have a disease in the DNA, you can easily detect that with DNA sequencing. You just have to do you have to take a sample off your patient, and then you can analyze that sample more quickly than before. You can analyze it quickly, and then after you analyze that sample, you can uh, tell based off of the changes in the DNA, you can tell what disease they have more easily. So this, pretty important. You probably want to know what disease you have. And then there's also genetic engineering in agriculture. So there's some pretty important genetic engineering things that you can do. Biotechnology, it's helped a lot with agriculture, and it lets you to essentially edit crops or livestock to make them, one, you can make them more resistant, or two, you can make them more efficient. So more recently, there's been a lot of diseases to crops that are pretty dangerous, like tobacco mosaic disease, etc. These can be really harmful to your crops and everything. So you wouldn't want this. And one way you can use to fix these diseases is genetic engineering. So you can instead of losing your entire crop, you can change the crop so that its DNA is more resilient to changes, and so that it won't be affected as much by these diseases. And this uh, allows you to essentially not lose as much crops to your diseases. And another way this helps with agriculture is with for example, livestock. So you can use DNA or genetic engineering to change certain genes. Like for example, you can use CRISPR Cas9 to insert certain nucleotides. And what this does is you could change a bunch of different genes, because genes code for everything. You could use it to make cows grow more meat, or you can use it to make cows more resilient to certain diseases. So all of this is pretty good stuff. And another way biotechnology, all this stuff is important, is in forensic science. So, I don't know a lot about this, but there's, when a murder is committed, for example, the murderer could bleed, it'll leave blood behind. You can take the blood, and then if there's not a lot of blood, that's still fine. But you can take the blood, you can amplify it with either DNA cloning or PCR, and then you can essentially read the blood's genome, and you would essentially know the DNA of the organism or the person who committed the crime. And you could check it with an online database, which does exist, or you could check it with an online database, or you could just take a DNA sample out of someone and then check that specific sample. Another thing that's important is environmental conservation. So there's two ways you could kind of look at this. One is you could use it for research. Two, you could use it for potential cloning, things like that. So one thing you could do is you could do species identification with this. You could see, for example, what species live in certain areas, so you can tailor your environmental conservation, movements, things like that, more for the species that live in that area. So essentially, there's a lot of online databases with DNA of a lot of different organisms. If you find the feces, the poop, or the fur of an animal left behind, you could test that. You could test the DNA in the fur or the feces or whatever, and you could compare to the online database to see what species live in that area, and you can maybe see if they're extinct or not, or you can see if they even do exist in the area anymore. You could also use it for cloning. So the way cloning works I don't have a slide on it or anything, but you can take the DNA of an organism, put it in the egg of another organism, and then use that to make an artificial organism without anything going on between the two organisms. And this helps with, like, for example, the white rhinos. They've been going extinct recently, and people have been trying to use cloning to bring them back or to make babies. And then another thing that helps that you could do with DNA is you could make biopharmaceutical or like drugs. And essentially, you can make more effective, more affordable treatments such as insulin, vaccine, and monoclonal antibodies. So instead of like making a new batch of your disease, instead of making a new batch of your drug or whatever every time, if it includes like live organisms, if it's like DNA or anything, you could quickly do that instead of extracting it from the organism. You could just clone it. And another thing is just scientific research and discovery. So it's pretty self-explanatory. DNA tools have helped a lot with different like research initiatives across a bunch of different disciplines. You could change the DNA of organisms now, so you could test. You can essentially, edit organisms, make them better. You could make them like do something that you want, and you could also essentially look at the DNA of something and then compare it with a normal piece of DNA, and then you would see the difference, and then that would be something you could treat, or you could just, look at irregularities in different DNA structures, and then you could find a pattern there, and then you could develop a diagnostic to a disease. And then lastly, even though DNA technology, all this stuff is pretty great, you have to be, you have to kind of know what's okay to do and what's not okay to do. So even though, it'd be pretty great if like all this biotechnology, we just like went full force with it. We just, did as much as we could with it. It might be good not to, because we need to sort of know like what is too much. So like it's probably not okay to take someone's DNA without their permission, or it's also good to make sure DNA is accessible to everybody, where we have DNA samples from every like ethnic group, every like racial group, so we could test them and use them. And it's important to just, be responsible with DNA engineering and not to, do everything you want with it. It's important to just be responsible and to ask yourself like what's okay, what's not okay. And yeah, this should be all. All right. Thank you. All right. I think if no one has any questions or anything, you yeah, should be finished. And we should be done with the class. So you guys can leave. Bye-bye.